to study role of senescence in aging, regulation of presence of senescent cells, how they accumulate in our organism with age, why they accumulate, and how we can tackle these mechanisms that regulate the accumulation of senescent cells in order to extend health span. So this is from the older review of hallmarks of aging, and senescence is one of the hallmarks. And what we learned over the years about senescence and senescent cells is that senescent cells accumulate in organism over the age, and together with this, there is increased risk of development of age-related pathologies. We have learned that immune system tries to control presence of senescent cells, and we studied this a lot, and I will mention a few things about this. But in the last decade, we also learned that if we eliminate senescent cells using genetic models, that we can extend the lifespan of mice by about 25 to 30%, and also extend the health span of mice. So what are these senescent cells, why they are formed, how it's possible that something that is damaging to the organism is formed naturally in the organism? So the idea is that senescent cells are formed as a protective mechanism, mainly as a result of activation of the oncogenes in the response of DNA damage in all other types of cellular stress. And the short-term activation of senescent cells is protective, and it's protective in many ways. It leads to tumor suppression, because if senescence is a terminal cell cycle arrest, and if we induce this terminal cell cycle arrest in cancer cells, this is tumor protective. We have demonstrated over a decade ago that senescence limits tissue damage, and it's kind of easy to understand in a model that if we have a cut in our skin, that actually fibroblasts that are frost cells to proliferate and to close the wound. But at the end, we want these fibroblasts to go away, because if they will continue to be there and proliferate, we will have a huge scar from every small cut. This is not what happens. What happens is that these cells, they enter senescence and they are eliminated by the immune system, and it happens in many different organs. So senescence is a protective mechanism in the short term, and it also plays a role during embryonic development, something that we were one of the labs that discovered it, and it's in transient guiding structures during development, and we worked on the placenta. But our kind of topic of today's conference is aging, and what happens with aging, that senescent cells accumulate, and once they accumulate, they secrete so-called SAS, senescence-associated secretory phenotype, that contributes to inflammation, and we think that senescent cells can promote tumorigenesis in their environment and can promote tissue aging. So how can we study it, or how we can understand why they accumulate in the tissue? And over the years, we have seen that immune system tries to uncouple positive short-term responses from the negative long-term responses. So senescent cells do need to be formed, but organism tries to regulate the accumulation by the immune system when immune cells, different types, control the presence of senescent cells and basically eliminate it. During the years, we had an idea already more than 10 years ago that there is a possibility to kind of mimic the ability of immune system and develop methods for directed elimination of senescent cells, which are nowadays called senolytic approaches. And we have discovered a couple of such pathways. One, about more than 10 years ago, it's overexpression of BCL2 family members in senescent cells that can be targeted by inhibitors, which were already known at this time. And use of this kind of inhibitors was shown in the last decade to be able to eliminate senescent cells in different diseases and conditions. So with this, we can look what are the properties of senescent cells and look at different diseases where senescent cells accumulate over the age. But I think with what we can kind of remember, that senescent cells bear persistent DNA damage response. They activate cell cycle arrest machinery. They are resistant to apoptosis because of the upregulation of BCL2 family members, and they actively interact with the immune system by the secretion of SASP and by calling the immune system, also using this SASP-secreted cytokines in order to eliminate senescent cells. And they can accumulate in many different diseases, and probably some of these mechanisms of control of their presence are disrupted during these diseases, and we can look further into how immune system actually controls senescent cells in different diseases and condition and in aging in general, and how we can activate the immune system even more in order to clear senescent cells. But back to the kind of scientific questions, how the senescent cells can affect tumorigenesis and tissue aging, and now using tools of directed elimination of senolysis, we can ask if senescent cells affect tumorigenesis or affect aging. And we have done it in different mouse models. So in one of the recent studies, we asked, okay, we know that senescent cells have a protective role. This is a tumor suppressor mechanism. So it's known, see, over more than 15 years that senescent cells accumulate in premalignant lesions. So in, let's say, one cell express oncogenic mutation, it goes over a few rounds on proliferation and then enters senescence, and these cells accumulate in premalignant lesions. But these lesions can be for years and decades in our body, but then something can happen and cancer develop, and it's known that they can develop from premalignant lesions. So we asked what would happen if we eliminate senescent cells from these premalignant lesions, and we have done it in the model of pancreatic cancer in mice. So we used BCL2 family inhibitors in order to eliminate senescent cells from premalignant lesions, and ask what would happen month and month later in the mouse models in terms of development of tumor. And we have found that this treatment and the elimination of senescent cells from premalignant lesions blocked cancer development later on, and the reference is down. So using the similar tools, we could ask what is the effect of senescent cells on aging, and how, and if we can use these tools to counteract problems with the immune system that lead to increased accumulation of senescent cells. So we used a mouse model where there is increase in accumulation of senescent cells due to the problem in the gene that are responsible for their immune clearance. And we have seen increased accumulation of senescent cells. And these mice die early and develop different age-related pathologies. And we could use, again, BCL2 family inhibitors to counteract this effect, both in mice that have normal aging and a mouse model of accelerated aging. And we have seen that we can extend lifespan and health span of these mice using elimination of senescent cells by a senolytic approach. Okay, so now with this in mind, we can ask more for general questions about accumulation of senescent cells in the organism. Okay. In the young, we know that there are senescent cells normally present in tissues in very low level because tissues are heterogeneous. We are all exposed to all kind of stress, oxidative stress from the air, from this kind of radiation from the sun, and so on and so forth. So senescent cells are formed and they are cleared by the immune system in order to keep the tissues in normal state of homeostasis. So one of the ideas was that senescent cells actually have a turnover. It's not that just simple accumulation. there is formation and clearance all the time, and there is a turnover. And we studied this question together with the lab of Uri Alon in our department, system biology lab, and we identified mathematical rules that we can understand how senescence, or can mathematically describe the half-life of senescent cells and their accumulation in the organism. So the reference is here. So what happens now with aging? One of the kind of ideas that came from this mathematical model work was that senescent cells inhibit their own removal by immune system. But there was no biological or molecular mechanism at the time to understand why this happens. So we used different models. While using different models to understand the role of senescent cells in aging, we recently discovered that this turnover, yes. So we can control the turnover using PD-L1. And why? Using anti-PD-L1 antibody. And why? Because we have observed that senescent cells upregulate PD-L1, and the regulation of PD-L1 is by a basic molecular, or connected to basic molecular mechanism of senescence, P16, which is a CDK inhibitor protein. And we have shown that P16 regulates protein stability of PD-L1 protein, and that we can use anti-PD-L1 antibody, and they are all famous about activating PD-L1 antibody, and they're, in cancer treatment, there are now many anti-PD-L1 antibodies. Some are blocking and some are activating. So we used activating PD-L1 antibody, meaning the antibody that not only blocks the signaling between PD-L1 and PD-1, but brings the immune system to attack cells that express PD-L1. And we were able to eliminate senescent cells, in this case, from the lung of mice. And this provides one example of a mechanism how senescent cells regulate their own removal, and how senescent cells accumulate with age. And we also have seen that senescent cells, there is a difference between senescent cells in the young organism and in the old organism. And there is increase in expression of some proteins, immunomodulatory proteins with aging, including PD-L1, which expression is increased with aging. So this is with a general model that we are trying to kind of look around, or trying to understand the regulation of senescent cells in the young organism. There is a normal turnover of senescent cells because we are exposed to different types of stress and damage, and senescent cells can be formed and they are cleared by the immune system. But if there is a disbalance for whatever reasons, and this is what happens with aging, and increase accumulation of senescent cells increases, there is inflammation from senescent cells and probably from other sources, which is known to increase with aging, and this might lead to accumulation of senescent cells and reduction in their immune surveillance, and therefore kind of further increase in the presence of senescent cells, which support the development of different age-related diseases, and this is only a short list of this. And we can think about targeting these senescent cells by several different ways. First, by senolytics, as I mentioned, then by specific antibodies against senescent cells, because there are, or might be, specific markers, and there are known specific markers on senescent cells that can be used in order to bring the immune system to clear the senescent cells in a safe way, and at the time when it's really necessary in order to reduce possible side effects from the general senolytic approaches, because we need to remember that senescent cells are essential in the short term, and therefore targeting them all over the body or in any situation might not always be beneficial, or sometimes can be dangerous. So with this, I would like to thank my lab and the funding sources and you for your attention.
Do you have results for maximum life extension in mice, real results?
Yes, so there are genetic studies and there are pharmacological studies with senolytics. With genetic studies, well-known papers by Jan van Duursen and colleagues, it's 25 to 27% life extension in mice. And with the senolytic approach in our study that I just mentioned during the talk, this were not in normal mice, but in the mice where they live shorter because of increased accumulation of senescent cells, we have seen similar increase in lifespan following treatment with senolytic drugs.
But maybe my point is always to take, let's say, sick mice and cure them, but it would be better with normal mice.
So that's
why don't you take
this mice
and let them die?
So that's I think there is sometimes in a mouse model you induce a disease specifically, right? But in the mice that I mentioned, your senescent cells were eliminated genetically, or in our model where it was just a problem with one immunosurveillance gene. That's not that they develop specific disease. We have shown they develop all variety of different diseases like any old mice or old humans develop. So this is different from looking at a specific disease. This is looking at a model that develop all variety of diseases and conditions that resemble aging phenotypes. And then these are delayed. This model is just a model, right? So by using the models, I think we accelerate discovery of things that are relevant to humans.
Just one question. Is it possible to have like finding out the level of a normal person or certain ages that you'd say, these are how many senescent cells you need, and if you go above a certain burden, maybe this person should be treated, and then you have a way to monitor it?
Yeah, so this is very, very central question in the field that you raise. It's super relevant question, and we study it in details, and other labs also studied it. So how we can monitor, right, presence of senescent cells in the organism. So that there are many companies that develop senolytic drugs around the world, but there is no single marker that we can use, a biomarker, right, to know how many senescent cells there are in every person, right? And this is definitely a very strong need to do this, right? Because in mice, it's easy. We can look at their tissues and identify the number of cells, but we, at the end, right, we all would like to eliminate senescent cells from humans, and therefore we need a biomarker that would allow us to know how many senescent cells are in every person, and that might be, as you mentioned, an indication for senolytic treatment or a readout of efficiency of such a treatment.